This is lecture 3 on the immune system, our last lecture on chapter 42, and we're going to study antibodies. So antibodies all belong to a class of proteins called immunoglobulins, and we abbreviate those as capital I lowercase g. And so this is an example of a antibody. And so they consist of two chains, two types of chains, the heavy chains, these are the heavy chains, and the light chains. And each of these have an area which we consider the constant region. So this light blue and this light green are constant, and the same on the other side here. And so these are going to be the same for the different classes of immunoglobulins. So if it's IgGs, all of the IgGs are going to have the same structure here. But what will vary is the variable region. And so this is the variable region of the heavy chain in dark blue, the variable region of the light chain in dark green. And so this is the antigen binding site. So this is going to be specific to a specific antigen. And that's what makes them specific to whatever particle or pathogen or protein your body is fighting off. You don't need to know the names of these anymore, but just know that generally they have this Y shape. They can form a pentamer here or a dimer here. So if you see them in those shapes, just understand. But they all have the same Y, and whatever specific variable site they have here, it's going to be the same here and here and here, and the same for this one, all of the different places. So whatever is on this side, whatever the binding is, it's going to be exactly the same as on the other side. Okay, your body has genes that code for these different regions, and basically what happens is you get a splicing together of different genes, and that's what gives you that variable region. So here we have these variable genes, which they call them V12 all the way up to V100, and then these diversity genes, which is D1 through D30, and then these joining regions, and so this is a J1 through J6. And then here's the genes coding for the constant region. So what happens is 1V, 1D, and 1G get randomly put together. So one of these 1 through 100s, one of these 1 through 30s, one of these 1 through 6 spliced together along with this constant region. And that's what gives you that variability, which is so important. And this just shows you kind of how that happens. So here we have our embryonic DNA, and this is that variable region with the V genes, the D genes, and the J genes, and here's our C's. And so this is going to determine here which class of immunoglobulin we have, and this is going to determine the antigen binding region of that. Okay, here's another picture I think is a little clearer. So here between one of these V's, and remember there's over 100 of these different V's, are going to get joined together with a J, and depending on the type of whether antigen, we would have the D as well. And so here we have we have the V2, the J, and the C, and then we get splicing, and we're left with just those 3 segments. So which 3 segments get put together is going to give you that variation. That's going to give you that variable region, that binding site. And we will talk about that in more detail, and it'll make more sense to you when we do protein synthesis, which we have not done yet. Okay, so how antibodies kind of work, which I think is the most important aspect of this lecture. So they can inactivate antigens in lots of different ways. So here we have a virus. Remember, viruses bind specifically to their host cells, and so that antibody can bind to those binding sites for the virus, block all of them so that virus can't bind to its host cell and attack it. It can do the same kind of thing to the bacteria, right? Anything where it might attach, it's going to bind to that and kind of block it. And then it can also bind to many different bacteria like this and cause this agglutination or this cluster. The other thing that it can do is, let's say it's a toxin or something in your body and it's soluble. So it's not a particle that your antibodies or your cells can see. And so here the antibodies bind to these particles and make them insoluble, so they come out of solution, they precipitate, and then then phagocytes can see them. So once these things are coated in these antibodies like this, or they're clustered together like this, or maybe you've brought it out of solution, now those phagocytic cells, those macrophages, can now find those and engulf them. So that's going to help in that process. The other thing that they can do is, we saw this in the beginning, I think in the first lecture, where they can activate that complement system. And so here the antibodies bind, and that activates those proteins to start forming that cluster, and then they form that pore in the cell, which leads to that MAC attack, or that cell lysis that we saw earlier. Okay, one of the important things about the immune system is that you have a memory. And so here we see kind of a graph of that. So here we have how much antibody you have in your blood, and here's the time. Okay, so you get exposed to some kind of antigen here, and so it takes a few days of incubation for that particular antigen to start to reproduce, so it's in high enough quantities, whatever, get to its location, whatever, and so now your body starts to see that, so it becomes under your body's surveillance, and your body starts to produce antibodies, and you're feeling sick, and then your body starts to clear it, you're starting to feel better, and you clear your infection, and your antibody levels will go down. So now, if you get a secondary infection, so the same antigen, antigen A, that you were exposed to here, you get exposed to it again. Well, now your immune system is like bigger, badder, and better. So you're going to get a much faster response. The antibodies are going to increase to a much higher number than they had in the first infection. It's going to happen much more quickly, and they're actually better because the antibodies will actually bind more tightly to the antigens in the second response, and so then you will clear your infection much more quickly. However, if you get infected with another, here's our secondary exposure, another exposure, our first exposure of antigen B, your body has to start all over again because you don't have a memory for antigen B. So again, you're going to have that incubation period, your body finds it, antibodies start to be produced, you start to clear it, you'll start to feel better, and so on. And now if you get exposed again, you again will have a bigger, better response. Okay, and the last thing I want to talk about is vaccination. I just think this is kind of fascinating. But vaccination actually comes from the name vaca, or vaccinia, which means cow. And this is a picture, I think this is Edward Jenner. Anyways, Edward Jenner was the first person to come up with a vaccination. People realized that individuals who had gotten smallpox and survived smallpox had an immunity to it and wouldn't get infected again. It was also, people were aware that milkmaids tended to not get smallpox. They would get cowpox, which was a much milder infection, and they would, and I had a picture, I must have taken it out, of somebody with smallpox. They get horrible scarring on their face and all over their body, but it's very disfiguring in their face from these pox. Well, the milkmaids would get cowpox, which was a much milder infection, and then they seemed to be immune to the smallpox. And so Jenner decided to take a woman who had cowpox and inoculate I think it was an 8-year-old boy. And they did that by lancing the skin, taking the pustules from the pox and putting it into the wound from the child. And the child got cowpox. And they were sick from the cowpox, and then they recovered from the cowpox. And then he did it again with smallpox to the same child, and the child did not get sick. And so that was the first vaccination. And so then they developed this vaccinia, this vaccination for cowpox using smallpox. And smallpox has been eradicated as a result of his work. How he ever was able to do that, I don't know. We would obviously not be able to do that today. But this is a picture of a bunch of kids lined up to get their vaccinations. And that's the end of this lecture. I hope it helped.